Well, Nesreen, that was an excessively generous introduction. Thank you very much. I didn't deserve that at all. And thank you all for giving up your evenings to come and listen to me. So I gave this title sort of Great Leaders, Innovators and Entrepreneurs Will Solve the World's Greatest Challenges. So let me just kick off by, and we were just having a conversation a couple of minutes ago, sort of, you know, I'm a great believer that individuals can change the world. And I think I've shared with some of you before that sort of, if I think of people across the planet who have changed the world, you know, I think of Bill Gates. He changed the world when he set up Microsoft. And he's changing the world for the second time by giving away all his money. I think of Steve Jobs. He's changed the world. I think Mark Zuckerberg has changed the world. I think Elon Musk is changing the world. I think even Bezos has changed the world. I mean, he started in, 1999, just dropping books through letterboxes, etc. And look at the impact of Amazon, and look what it's doing to the high streets, even in this country, etc. He has changed the world. But you know, just listening to that, you think, well, why are all these 5 individuals in the US? Can we think of an individual like that in the UK? And I have to say I struggle. And here, when I talk about changing the world, I'm talking about individuals who have created new industries. They've created hundreds of thousands of jobs. They've created a lot of personal wealth. And they've created a lot of wealth for other individuals. I mean, Apple as a company, I don't know how many millionaires they produced. And they probably produced a few billionaires as well. You know, these are individuals who have changed the world. And so I do believe in the power of the individual. Now, I also think across the world, we're facing some massive global challenges. You know, like you all, I worry about climate change. And, you know, I feel a little powerless. I don't know what to do about it. I worry about pollution. I worry about energy. You know, in 20, 30 years' time, we're going to need 50% more food, 50% more clean drinking water. I don't know how we're going to get it across to 10 billion people across the world. These are major, major challenges. In my own space, I worry about healthcare, but importantly, I worry about more affordable medicines. You know, there's no point producing a new medicine if most people across the planet can't afford it. So I think we're facing many, many challenges. And I think the solutions to these challenges are new technologies. I'm a great believer in new technologies. And I know that's certainly sort of Bill Gates' philosophy. He thinks technology is the answer to everything. I should say, I don't know if you've seen, there's 3 programs on Netflix about Bill Gates. They're sort of half-hour long each. If you've not seen them, I strongly recommend them. I mean, I think the guy is an incredible individual. He's an absolute genius, you know, and what he's doing is just quite remarkable. I mean, it's remarkable to see that he will focus on a problem, he will talk to the world's experts about that problem, and very quickly he will be the world leader in that problem. And it's just astonishing. And Deborah, I think we should try and get Bill Gates an honorary degree in this university. I think he deserves it. But anyway, sort of, I'm going off tack a bit. So people is important. Technologies are important. I think collaborations. I don't believe in establishing a platform or setting up an assay or developing a technology if somebody else has got it. If somebody else has got it, I'd rather just collaborate with them because that's much, much faster. I also think many of these problems we're facing, you know, there's no way even a university like Oxford can tackle these problems. You know, I don't even think as a country we can tackle some of these problems. We're going to have to collaborate with other countries. We're going to have to create big international partnerships. That's going to have to be the way forward. And I think these sorts of individuals, they are going to have to demonstrate commercial acumen. And they'll be brilliant scientists and brilliant technologists. They're going to have to be great leaders of people. They're going to have a vision and a passion, etc., etc. But they're going to have commercial acumen and also political finesse. Because to tackle some of these complex issues requires all of those sorts of skills. Now, what I'll do in the next 40 odd minutes or so, I'll just share with you and try and illustrate in just sort of one area some of the challenges that we face. And the area that I know most about is healthcare and the production of new medicines, etc. So I hope some of the lessons that we might tease out from that are applicable in some of these other global challenges. So let's kick off and let's just think about, here I work in the #1 university in the world. The medical school's been #1 for the past 8 years. I'm focused on trying to generate new medicines for patients. Right? But let's just think, what do patients expect from people like me? What do patients' relatives expect? What does society expect? And what do healthcare providers expect from us medical researchers? I think what they want is they want more novel medicines. They want more effective medicines. They want more affordable medicines. And they want them quickly. And let me just take each of those in turn. Novel, first of all. Now, in the UK, I think our population is 65 million, something like this. In the next 12 months, in the UK, 350,000 people will get diagnosed with cancer. That is 1,000 people every day. That is one person every 90 seconds. And half of us in this room during our lifetime will have a diagnosis of cancer. In the next 12 months, 14 million people across the planet will get diagnosed with cancer. We desperately need new treatments. I was at an award ceremony on Tuesday evening in London, and they were collecting money for a teenage cancer charity. And the lady who was the CEO of this charity said, every day in the UK, 7 teenagers are diagnosed with cancer. And they got this young man who's now about 23, 24, and he told his story about how he was a student at Durham. He was getting ready for his finals. He felt a lump in his throat. He went to his GP several times. The GP told him to go away. There's nothing wrong. His sister was a doctor. She pushed him into going into the hospital. And there they did a scan and found out he's got cancer, etc. And he told his story. And then he said that he was admitted to hospital, and he was the youngest patient on the ward because the next person was 40 years older than him. And he talked about the challenges that he as a teenager felt having had cancer, etc. But 7 people every day in the UK. I mean, it's frightening. I worry a lot about dementia. And this is why 4 years ago we built a Dementia Institute here in Oxford. Now, let me just share some figures on that. So today, across the UK, we've got 850,000 people with dementia. In 2050, that number will be 2.1 million people with dementia. We will have 2 cities the size of Birmingham with dementia. The average cost of caring for those patients is £32,000 a year to the taxpayer. That's despite the fact that 2/3 of the costs are borne by the relatives. So if you add all that up, we are spending £26 billion a year just caring for our dementia patients. All of us who live to more than 80, 1 in 6 of us will have dementia. We've not come up with a new drug for dementia since 2002. That drug only works in the first 9 to 12 months. And then after, as the disease declines, it stops working. And this one disease is going to financially cripple many societies. And I don't think we're anywhere close to getting a drug that's going to slow down the progression of this disease. It's frightening. Aging, Brian and I are trying to set up this project on aging, and so is Deborah. But you know, in the next 20 years, the number of pensioners in this country with diabetes will go up by 120%, so more than double. The number of pensioners with cancer will go up by 180%. So that's almost a tripling in the next 20 years. AMR, antimicrobial resistance. I mean, Sally Davis, you know, for the past 5, 6, 7 years, when she was the chief medical officer, she really publicized that this is a global crisis. We desperately need a new generation of antibiotics. So apparently at the moment, 700,000 people a year are dying because they are resistant to existing antibiotics. And it's estimated that in 2050, if we don't have a new generation of antibiotics, there will be 10 million people a year dying because they're resistant to antibiotics. And that will cost the globe 100 trillion loss in GDP, 100 trillion. These are figures that Jim O'Neill published about 3-4 years ago. I mean, these are frightening statistics. Mental health, you know, this is a massive issue right across the planet. So across Europe, it's estimated that 83 million people have got or had some mental health episode during their life. It's estimated that 20% of people will have a depressive episode during their life. And I heard a horrific figure recently that if you take 15-year-old girls in this country, out of every 150, one of them will be anorexic. Rare diseases. There are 7,000 rare diseases out there. 30% of those kids do not reach the age of 5. It can take 6 to 8 years to get a diagnosis for a rare disease. And 95% of those patients have absolutely no treatment. Let me share with you 3 anecdotes. And when these things happen, you never forget them. So in 2007, before I came back to Oxford, I was working at GSK. And I went to visit a elderly care home, and there was dementia patients in there, and I went to see one of these dementia patients in a side room. And the dementia patient, he didn't acknowledge, I think he was in his mid-70s or something like this. He didn't even acknowledge that I was in the room. His daughter was sitting with him, and I chatted to his daughter for about 5 minutes. At the end of this conversation, just as I was leaving, his daughter touched my hand and she said, "Please help my father." And my immediate reaction was, it's too late for your father. And the tragedy is, if that happened to me today, I would have exactly the same reaction. 12 years on, we have made no progress as far as a new treatment for slowing down the progression of Alzheimer's. 4 years ago, I gave a public lecture here in Oxford on one of our alumni weekends. It was on a Sunday morning. And at the end of this lecture, this lady comes up to me and she says, "Professor Bountra, my daughter died with a brain tumor earlier this year. And if you want to study her brain, this is where you can get it from." And she literally handed me a scrap of paper. And on this paper was her daughter's name, her date of birth, the day she died, and where her brain was stored. It was lunchtime after this, and my wife came to pick me up, and I said to my wife, I've just had a complete stranger come up to me and offer me the brain of her dead daughter. You know, this is what parents feel. About 3 years ago, a lady came to see me, and she lives in Bath. Her daughter had a rare GI stromal tumor. So this tumor is about 1 in a million, so there's probably about 60 individuals across the country with this tumor. So the daughter didn't come to see me, but her daughter's physician, who lives in Cambridge, he came. So the mother and the physician are sitting in my office, and the mother gets very emotional. And she's getting very emotional because there is no treatment for her daughter. The pharmaceutical industry is not trying to discover a treatment for her daughter. Her daughter is gradually dying, and she and the daughter and the family have absolutely no hope. I eventually said to the mother, I said, how's your daughter doing now? And she says, my daughter is 22 years old, and they've just removed the whole of her stomach. And I sat there thinking, how on earth does any parent cope with that? You know, at the time my daughter was about 25, and frankly, my daughter's my life. And I'm thinking, how does any parent cope with that? We need more novel medicines. We also need more effective medicines. And you might think, well, why is he saying effective? Aren't all the drugs that are out there effective? About 4 or 5 years ago, there was a publication where they looked at treatments for solid tumors. So at the time, we had 71 treatments for solid tumors. And in this publication, they looked to see how much increase in progression-free survival or overall survival these 71 drugs provided. And they found that the average increase was in progression-free survival was 2.5 months. The average increase in overall survival was 2.1 months. And they concluded in that study, of those 71 drugs, only 30 of them had clinically meaningful efficacy. So less than half the drugs that are out there had clinically meaningful efficacy. We need more affordable drugs. So again, a publication a few years ago, they looked at, in the UK, in the year 2000, we had 69 drugs for treating cancer. 50 of those were cytotoxic agents. The average duration of treatment was 181 days. The average cost of that treatment was £3,000. And in the year 2000, £3,000 equated to 20% of our GDP per capita. They did the same analysis a decade or so later, in 2013-14. And now there was a further 63 drugs for treating cancer. The average duration of treatment was now 263 days, so it's gone up from 181 to 263. The average cost of the treatment was now £35,000, so it's gone up from £3,000 to £35,000. And £35,000 equated to 140% GDP per capita, so it's gone up from 20% to 140%. And that trend is continuing. I was just talking to Nisreen earlier on. That some of you have probably heard about 6 months ago, Novartis had a drug approved for spinal muscular atrophy. This drug is a single infusion, and the cost of that single infusion is $2.125 million. Now, even in the US, many patients will not be able to afford it. The UK will never pay that much money. Now, I'm not criticizing Novartis, but for me, the lesson is that we have to drive down the cost of this platform by at least 1 or 2 orders of magnitude. So we need more affordable drugs. And we need them quickly, because if you're a patient, even tomorrow is too late. You want the drugs today. So novel drugs, effective drugs, affordable drugs, and quickly. The problem with the way we're doing drug discovery at the moment, frankly, it's too costly, it's too risky, and it's too slow. Again, let me just take each of those in turn. So around about 2012, Forbes did an analysis where they looked at a number of pharmaceutical companies. They looked to see how much money they spent on R&D in a finite period. And they looked to see how many drugs they launched in that period and they divided one number by the other and came up with an average cost of a new drug. In that analysis, their conclusion was that AstraZeneca, the average cost was $11.5 billion. So for one drug, $11.5 billion. Amgen, who were the best in that analysis, even there, the average cost was $3.5 billion. And many of those drugs were not truly novel drugs. They were what we call me toos or new formulations. And what we need is truly novel drugs. So it's too costly. I think everybody recognizes today that the average cost of a new drug is somewhere between $3 and $4 billion. Just to put into context, I don't know the exact figure, but I suspect the average cost for running this university, including all the colleges, is probably about £2.5 billion a year. So the average cost of a new drug is comparable to that. And you just imagine how many people are employed in this university. So it's too costly. It's also too risky. So again, a publication a few years ago, they looked to see, I think it was in 2003, there was 529 molecules in development for cancer. So they were either in phase 1 or phase 2 or phase 3. 529. And they looked to see a decade later what happened to those 529 molecules. And they found that 45 of them had made it to the market. Well, that's great. 95 were still in development. But 389 molecules were terminated. So as a community, as a biomedical community, we generated 389 molecules and then took them into the clinic and then terminated them. Can you imagine how much money we spent on those 389 molecules? Can you imagine how many people's careers went into those 389 molecules? And can you imagine how many patients were exposed to those molecules and they didn't deliver a new treatment? So it's too risky. In that same study, they looked at if a molecule is in phase 1 for cancer, what's the probability it will make it to the market? The answer was 7.5%. Only less than 1 in 10 molecules in phase 1 for cancer makes it to the market. And they looked to see if a molecule is in phase 3 for cancer, so these are the final registration studies just before you launch the drug. If something's in phase 3, what's the probability it will get to the market? The answer was 33%. So 1 in 3 molecules in phase 3 makes it to the market. So the process is too risky. It's also too slow. And a couple of my colleagues wrote a paper a few years ago where they looked at when did we come up with some data that a particular target would be useful in cancer, and then how long did it take to translate that idea into the clinic? And they estimated that it was anywhere between 6 years and 30 years, 30. The process is too costly, it's too risky, and it's too slow. So why? This industry employs some of the smartest people on the planet. They have access to great technologies. They can collaborate with whoever they want. Why is it so risky and costly and slow? I think there's a number of challenges. There are firstly scientific challenges, and there are other challenges that you might describe as being organizational challenges. So again, let me take those in turn. The scientific challenges. I think it's fair to say that most human diseases, we do not really understand these diseases at the molecular level. Patients are incredibly heterogeneous. We talk about Alzheimer's patients, but I guarantee if we took 100 Alzheimer's patients, some of those patients may have forgotten where they left their key. Others will not recognize their own children. Some will be aggressive, some will be depressed, some will be agitated, some will be anxious, some will be, lots of different symptoms. These diseases are incredibly heterogeneous, and it's very difficult in most situations to pick one patient and say, these are the molecular causes of your phenotype or your symptoms or your disease. We also, many of us use them, animal models. I do not believe that animal models fully recapitulate human disease. We will never ever have an animal model of Alzheimer's disease or schizophrenia or depression, etc. For many diseases, we don't have good biomarkers. So these are readouts that we can use in the clinic to assess efficacy of new molecules. So if you're doing a study in Alzheimer's, you can't say to the patient, oh, is your memory better today than it was last month? Or a depressed patient, or are you less depressed today than you were last week? We need better biomarkers. And it's fair to say there are drugs out there, we don't know how they work at the molecular level. So paracetamol, acetaminophen, we've all taken it. Probably 100 million people on the planet today took paracetamol, and we do not know how paracetamol works. So if you don't know how existing drugs work, how can you design better ones? So there are major scientific technical challenges. There are also organizational concerns. One of the biggest worries I have is that a lot of academics, a lot of biotech companies, and a lot of pharma companies, they all work on the same few ideas. And the reason they all work on the same few ideas is because they all read the same literature, and they go to the same conferences, they talk to the same opinion leaders. So they go back to their respective labs, and then for the next 5, 6, 7 years, they work in parallel, in secret, to come up with a proprietary molecule. And then they take it into the clinic, and we know when we test novel molecules for novel targets in patients for the first time, the failure rate is 70, 80, 90%. I've heard figures as high as 95%. Most ideas do not translate from the lab into the clinic. So you can imagine if you've got 20 companies doing exactly the same thing, if one of them fails, the other 19 are going to fail as well. This is a horrendous waste of money. It's a waste of people's careers, but importantly, it's a massive waste of patience. And you could argue that ethically and morally it's wrong. The way we're doing drug discovery today, we are exposing patients to molecules that other groups already know are destined for failure. The other challenge we've got is that when I talk to many of my industry colleagues, they say that 50, 60, 70% of the time, they cannot reproduce academic literature. So there's no point us getting defensive about it. We need to do something about it. So lots of challenges. Now, of course, I've painted a pretty bleak picture. I've identified lots of problems and challenges and so on. Now, it's easy to identify problems. The tough thing is coming up with solutions. So let me share with you what we've been trying to do in Oxford for the past 14, 15 years. And I think I can probably sum it up in sort of 4 ways. Firstly, we've tried to pool resources and share risk. So currently, we are working with 9 large pharmaceutical companies. And each of these pharmaceutical companies has given us 5 million euros of funding over 5 years. So we have more than 40 million euros of private funding. We've got funding now from 7 different patient organizations. So Alzheimer's Research UK, 4 years ago, they gave us £10 million, and then a couple of months ago, they've given us another £12 million. And we've got funding from rare disease charities, etc., etc. The Wellcome Trust, to date, have put close to £60 million into our lab here in Oxford. So we're pooling all of these resources to share risk. The second thing we do is that we've decided that we're only going to work on completely novel ideas and novel targets. We're not going to work on ideas and targets where there's already 100 publications and 50 labs working on it. We want to work on completely novel ideas. And we deliberately work on ideas that people think are impossible, undruggable, intractable. Because I believe our job in academia is to drive innovation. So what we do is we work on completely novel genes or novel proteins. And for those, we generate very high quality tools. So we purify the human protein. I'm not interested in rats and mice. We build assays for that protein. We work out the three-dimensional structure of that protein. We generate small molecule inhibitors for that protein, and we generate antibodies for that protein. These tools are very high quality, not because we're clever. They're high quality because we're working with these 9 large pharmaceutical companies. And through that, we get access to their expertise in medicinal chemistry, or their compound collections, or their screening capabilities, or how we develop cellular assays, etc. So we work on novel genes and we generate novel tools, and by doing that, we're trying to drive innovation. The third thing we do, and this is probably what makes us most unique, all of these tools, we make them freely available. We don't take out any intellectual property, we don't take out any patents. We give them away freely to anybody in academia, anybody in biotech, and anybody in pharma, because we believe that's the best thing we can do to accelerate science and therefore accelerate drug discovery. So you can imagine every academic who comes into our office wants to collaborate with us, because they know we've got no secrets, we'll share all of our know-how, all of our expertise, and all of our reagents. And that transparency creates a lot of trust, which is great for collaboration, it's great for science, and it's great for drug discovery. So we're now collaborating with more than 300 academic labs all over the world. And so they take these novel inhibitors or antibodies, and then they can test them in whatever they like. It could be a cancer model, a model of inflammatory disease, some neuropsychiatric disease, some rare disease, it doesn't matter. And of course, academics, all they care about is publishing. So they then publish this data. So this is a way of getting global academia to take our novel, high-quality tools and generate lots of new biology. We're crowdsourcing science. And by crowdsourcing science, we're de-risking these novel genes and novel targets. And the fourth thing we do is that all of our data, all of our knowledge, all of our reagents, we release it to the world immediately. We don't sit on it for 12 months while we're writing up our manuscript, because in those 12 months, there'll be other people outside in other labs doing something that we've already done. And that's a waste of money, it's a waste of resource, it's a waste of time. So by releasing everything immediately, we're trying to reduce duplication and we're trying to reduce wastage. So 4 things. Pool resources to share risk, work in novel, impossible areas to generate novel tools to drive innovation, make everything freely available to crowdsource science, and release everything immediately to reduce duplication and wastage. So what's been the impact of this? So we started generating novel inhibitors back in 2009. And we decided that we would work on epigenetic proteins. So we deliberately chose epigenetic proteins because these proteins mediate the effects of environment. So the environment could be things like stress, diet, exercise, sleep, pollution. And increasingly we recognize that environmental influences have a massive effect on the initiation, the development, and the maintenance of many of these diseases. Cancer, inflammatory diseases, neuropsychiatric diseases, etc. Now within epigenetic proteins, we decided to focus on a couple of families of proteins. The names don't really matter. But one of these protein families is called bromodomains. Now when we started this program in 2008-9, Many people said we were crazy because these proteins are undruggable. They're intractable. It's a protein-protein interaction. Well, we decided to work on them. And then in 2009-10, we generated our first inhibitor for one of these proteins. The protein was called BRD4. It's bromodomain 4. In humans, there's 61 of these bromodomains. And this was an inhibitor for BRD4. And we generated this inhibitor, and this inhibitor, it was potent at BRD4. It was selective. And also it was cell active, so it got across the cell membrane and bound to the protein. When we looked in the literature at the time, there was a handful of publications on BRD4. And one of these publications said that when BRD4 is fused with another protein called NUT, those patients develop a rare cancer called NUT midline carcinoma. It's such a rare cancer that most oncologists can't even diagnose it. Now, when we generated this inhibitor, we worked closely with GSK and we worked closely with Jay Bradner. Jay Bradner is a clinical oncologist. He was at the Dana-Farber. He's now the head of research at Novartis. So we worked together to generate this inhibitor. Now, Jay had access to patients with NUT midline carcinoma. So what he did was he took cancer cells from these patients and he put this inhibitor on top of those cells. And he showed that the inhibitor stopped the cells from proliferating, stopped the cells from multiplying. Of course, that's exactly what you want in an anti-cancer drug. He also showed that the inhibitor killed the cancer cells, caused apoptosis. And then when he took these patient-derived cancer cells and he put them into a mouse, the cells multiplied, the tumor got bigger and bigger, and when he dosed the compound, it stopped the tumor growing. Now, we published all of that data in Nature in December 2010. It's instructive to think what's happened in the past 9 years. So that molecule that we generated, we, GSK, and Jay Bradner have given out to well over 1,000 labs across the world. We've also made that inhibitor available to chemical suppliers like Sigma and Aldrich, so people can buy it directly from those companies, and they've probably sold that compound to several thousand labs across the world. As a consequence of this, today there are well over 1,000 publications on BRD4, many of them using this inhibitor. And the data in those publications suggest that this target, this inhibitor, is useful in a whole range of other cancers. There's even a publication in sepsis, in fibrosis, in cardiac hypertrophy, and even a publication in male contraception. And there's also probably others that I don't even know about. This is crowdsourcing science. As a consequence of these publications, many pharma companies have started their own proprietary program, so to generate their own molecule which they can patent. And today, I understand there are more than 35 clinical trials underway with BRD4 inhibitors from several tens of companies, pharma companies, biotech companies. So we've accelerated academic science, we've enabled proprietary programs in pharma, we've catalyzed translational studies in patients. But on top of that, Jay Bradner, in 2011, secured $15 million of VC funding to set up a biotech in Boston to take this target all the way into the clinic. The company was called Tensho Therapeutics. And then in January 2016, Tensho Therapeutics was bought by Roche for $535 million, so more than $500 million. So by releasing these novel inhibitors, we're also enabling the creation of new companies and new jobs. Now that was the very first inhibitor we generated. And since then, we've generated well over 70 inhibitors, and they're all freely available to the world to do with whatever. So that's the impact. What have we been doing more recently? So 4 years ago, we went back to the Wellcome Trust, and we said we now wanted to work with clinicians, geneticists, disease experts. And we were going to work with these individuals and get them to help us identify novel genes which they thought could be drug targets of the future. And for these genes, we would do what we're good at. And what we're good at is making the protein, building assays, working out the structure of that protein, and generating chemical starting points. This package of reagents we call the target enabling package, because it would enable that target both in academia but also in industry. Now, we said over 5 years that we would generate 21 of these target enabling packages. We've just finished year 4 and we've already done 30. It's likely that we will probably deliver 40 at the end of the grant. Now, these target enabling packages we've done roughly in 4 broad areas: in inflammatory diseases, metabolic diseases, cancer, and neuropsychiatric diseases. In each of these 4 areas, what we've done is created these small committees. There are about 25 to 30 people on each committee. Half of them are from academia, the other half are from industry. And these individuals have been giving up their time for free to help us identify novel genes which they think could be drug targets of the future. So Kay Davis here in Oxford chairs our neuropsychiatric committee, Julian Knight, the inflammation, Steve O'Reilly in Cambridge, the metabolic, and Paul Workman in London, the oncology. So that's target enabling packages. The other thing we've done as part of this program, a couple of my colleagues, Frank von Delft and Paul Brennan, brilliant scientists, they've developed a new platform which they call XChem. So this platform basically is designed to help us generate binders for undruggable proteins. So if you envisage maybe some proteins have got a very flat surface and it's hard to get something to bind, as opposed to if it was a pocket, then you can design inhibitors that fit into the pocket. Now, there are many proteins that when we use biophysical, biochemical screens, we can't identify low affinity binders. But what Frank and Paul have done is develop this platform where they are screening libraries of compounds using crystallography. And now they've ran several tens of screens and they've identified low affinity binders for many such proteins. At the moment, industry and academic groups are all over them. They're collaborating with so many people across the world to generate low affinity binders. And now we're trying to optimize these into molecules that potentially in time we could take into the clinic. What we've also been doing more recently is building links with patient groups. And of course, patient groups are very powerful today. They're powerful because they can tell governments where to spend their research dollars. But we want to work with patient groups for 2 reasons. One, because we want their help in accessing primary cells or tissue from patients. Because I think that's a much better way to come up with new targets for diseases. So just like we can get cancer cells or immune cells from patients, and then we can put our inhibitors on top of these cells to see what they do. But we also, when we generate a novel inhibitor, and let's say we imagine it might be useful in Huntington's disease, well, I don't know who the world experts are in Huntington's, but the Huntington's charity does. So we can give them the inhibitor, then they can disseminate it across the world. What we've also been doing more recently is trying to build 2 major projects. One is a national project, and the other is an international project. Let me just describe each of those in turn. So the national project is the following. We all appreciate that elderly patients don't have one disease, they normally have half a dozen diseases. Maybe a bit of osteoarthritis, a bit of diabetes, cardiovascular dysfunction, renal dysfunction, a bit of dementia, etc., etc. We now believe there are pathways or targets that we can go after which will affect multiple morbidity associated with aging. So with Brian's help and Deborah's help, we're building this national effort, working with the University of Dundee, the Medicines Discovery Catapult in Manchester, the University of Birmingham, ourselves in Oxford and the Crick. And we want to work together to take some of these novel targets, generate clinical molecules, and test them in patients in the NHS. So it's a national project to come up with new therapeutics for aging. We're also trying to build a major international effort. So you're all aware that in each of us there are 20,000 genes or 20,000 proteins. I'm being shown the watch. 20,000 genes and 20,000 proteins. Now, if you look at all the drugs that are out there, they probably hit about 1,000 of these proteins. If you look at all the targets that everybody's working on at the moment, it's probably, let's say, another 2,000. There's probably about 14,000 genes that we've not even touched. We don't have any tools. We'd have no proteins, no inhibitors, no antibodies, no structures, no assays. So we're having a conversation with the Wellcome Trust. How can we create an international effort basically to generate tools for the whole of the human genome? So this is about translating the human genome. Now, of course, to do that, we can't do it on our own. You know, if we just carry on at the rate we're generating TEPs, et cetera, it'll take us another 100 years, et cetera. So we're thinking about which major centers across the world we can partner with, which major funders across the world can we partner with, and importantly, we're going to have to develop some new technologies which increase our throughput by at least 1 or 2 orders of magnitude. It's the only way we're going to do it in a reasonable time frame. So that's the conversation we're having with Wellcome at the moment. So Nazreen, apologies, to sum up then. I believe that in early discovery, there is too much secrecy, too much competition, too much duplication, too much wastage. What we've been trying to do is we've been trying to bring together lots of clinicians, lots of academics, lots of pharma companies, lots of patient groups, also working with funders and regulators. We're trying to work together to come up with completely new targets for drug discovery. New clinically validated targets or de-risked targets for drug discovery. If we do this, we believe this will be good for industry, it'll be good for patients, it'll be good for society. We're also now working with social scientists and ethicists and lawyers and policymakers to think how we can apply this model to generate more medicines more quickly. What we're trying to do is to create a new ecosystem for drug discovery, an ecosystem that hopefully will deliver more novel medicines, more effective medicines, more quickly. And I hope, ladies and gentlemen, that they will also be more affordable. Thank you very much.